Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Today our topic is UV visible spectroscopy, which we continue with the part 4. Instrumentational design of UV visible spectrophotometer in single beam and double beam operators. We know that there are two types of UV visible spectrophotometer. One is known as single beam spectrophotometer and the other is known as double beam spectrophotometer. Instrumentation Instrumentation of UV spectrometer includes the monochromators, filters or dispersing device used to isolate and transmit a narrow beam of radiant energy from the incident light, which determines whether the instrument is classified as photometer or a spectrophotometer. Spectrophotometer used here detects the percentage transmittance of light radiation when light of certain intensity and frequency is passed through the sample. Both can be a single beam or a double beam optical system. Single beam spectrophotometers, light from the source is carried through lens or through aperture to pass through a suitable filter. The type of filter to be used is governed by the color of the solution. The sample solution to be analyzed is placed in cuvets. This is the main instrumentational diagram of single beam spectrophotometer. The light enters into the lens through the UV visible source through an entrance slit. Then it strikes with the objective lens, and with the help of different monochromators and gratings it passes through different type of slits and light is controlled. Then light passes through the exit slit and it transmit or absorb into the cuvette and strikes to the detector or phototube. The detector or phototube gives us the UV visible lambda max on the display screen. After passing through the solution, the light strikes the surface of detector. The detector can be barrier layer cell or phototube and produces electric current. The output of current is measured by the deflection of needle of light spot galvanometer or microammeter. This meter is calibrated in terms of transmittance as well as optical density. The readings of solution of both standard and unknown are recorded in optical density units after adjusting instrument to a reagent blank. Now we will see the working of UV visible spectrophotometer in a video.
Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
Double beam UV visible spectrophotometer includes two beams which are formed in the space by a U-shaped mirror called as beam splitter or beam chopper. Chopper is a device consisting of a circular disc. One third of the disc is opaque and one third is transparent. Remaining one third is mirrored. It splits the monochromatic beam of light into two beams of equal intensities. This is the flow diagram of UV visible spectrophotometer, which is used nowadays. It has mirror 1 from the light source, then the light passes through the slit 1 and through diffraction grating it passes through the slit 2, where there are some certain types of filters which we learned in previous lectures. From there the light strikes to different types of mirrors and then bounce back. One light passes through the reference beam or the reference sample and the other from the sample beam or sample covets. The detectors detects the transmittance and absorption of light both in the sample and in the reference blank cell. The intensities are recorded by the detector and displayed on the screen. This is a picture of double beam spectrophotometer, which is also available in pharmaceutical chemistry lab. What are the advantages of single and double beam spectrophotometer? Single beam spectrophotometer is simple in construction, it is easy to use and economical. Double beam facilitates rapid scanning over wide lambda or wavelength region. Fluctuation due to radiation source are minimized. It does not require adjustment of the transmittance at 0% and 100% at each wavelength. It gives ratio of intensities of sample and reference beams simultaneously. The disadvantages includes in single beam any fluctuation in the intensities of radiation source affects the absorption. Continuous spectrum is not obtained. While in double beam construction is complicated, instrument is quite expensive. This is the comparison of single beam instrument and the double beam instrument. Here also some points are included which we already learned in our previous UV visible spectrum parts. Here some advantages and disadvantages are also included. This slide contains some types of electronic transition in UV visible spectrum. If the transition is between sigma to sigma star pi orbitals, then this type of excitation is known as bonding. If it is from pi to pi star, then it is anti-bonding. If it is n to pi star, then it is non-bonding, and if it is n to n star, then it is also non-bonding. Different type of solvent effects includes in pi to pi star transitions. If there is a solvent which is polar in nature, it shows bathochromic shift, while in non-polar solvent it shows hypsochromic shift. In n to pi star transitions, in polar solvent it shows hypsochromic shift, while in non-polar it shows bathochromic shift. Some applications of UV visible spectrophotometers include it is used in the detection of concentration of substances, detection of impurities, structure elucidation of organic compounds, monitoring dissolved oxygen content in freshwater and marine ecosystem, characterization of proteins, detection of functional groups, respiratory gas analysis in hospitals, molecular weight determination of compounds. The visible and UV spectrophotometer may be used to identify classes of compounds in both the pure state and in biological preparation. This is the end of UV visible spectrum. These are the reference books. Thank you so much for attending this lecture.